Hello students, so we are doing the lesson Biomolecules. So dear children, today we are going to discuss nucleotides in this lesson. So nucleotides are basic unit of the nucleic acid. Now they also form energy carriers. Certain nucleotides act as the coenzymes also. Some nucleotide function as chemical messengers. A nucleotide is a condensation product of three chemicals: a pentose sugar, phosphoric acid, and a nitrogenous base. Three components form the nucleotide, that is pentose sugar, phosphate group or phosphoric acid, and nitrogenous bases. Two type of pentose sugar are present in the nucleotide. It can be ribose sugar or it can be deoxyribose. Ribose is C5H10O5 and deoxyribose is C5H10NO4. Both of them occur in the furanose state or the pentagon ring with one oxygen and four carbon atom. The fifth carbon having CH2OH complement lies outside the ring. The carbon atom 2 contain two hydrogen atom in the deoxyribose. It is hence called 2 carbon deoxyribose, and ribose differ from deoxyribose in having hydroxyl group OH group instead of a second hydrogen at the carbon 2. The deoxyribose occur only in those nucleotides which make up the hereditary material DNA, that is deoxyribonucleic acid. Ribose is found in the nucleotide forming the RNA, that is ribonucleic acid. And a number of other compounds like AMP, ATP, NADP, and then NAD, FAD, coenzyme A, etc. Nitrogen base are heterocyclic compound. They are of two types: substituted purines and substituted pyrimidines. Pyrimidines are 6-membered ring while the purines are 9-membered double ring. A pyrimidine ring has nitrogen at 1 and 3 position. A purine has the imidazole ring join at pyrimidine ring at 4 and 5 position. It has nitrogen at 1, 3 and 7 and 9 position. There are two types of purine, that is adenine and guanine. Pyrimidine, there are three types: cytosine, thymine, and uracil, represented by C, T and U respectively. Adenine is represented by A and guanine is represented by G. Now, combination of nitrogen base with the pentose sugar is known as nucleoside. Okay, if nitrogenous base and sugar, if two are present, then it forms the nucleoside, and if phosphate group is also added, then it will convert into the nucleotide. The nitrogen base combined with the sugar molecule at its carbon atom 1 in the glycosidic bond, that is C and C, by one of its nitrogen atom, usually 1 in the pyrimidine and 9 in the purine. Depending upon the type of pentose sugar, nucleosides are differentiated into ribonucleosides and deoxyribonucleosides. Now, uracil produce nucleoside with only ribose sugar. Thymine similarly form the nucleoside with only deoxyribose sugar. Other nitrogen base produce nucleoside with both ribose and deoxyribose sugar. Different nucleosides are adenosine, adenine plus ribose, deoxyadenosine, adenine plus deoxyribose, guanosine, that is guanine plus ribose, okay, then deoxyguanosine, then that is guanine plus deoxyribose, uridine, uracil plus ribose, then deoxythymidine, that is thymine plus deoxyribose, cytidine is cytosine plus ribose, and deoxycytidine is cytosine plus deoxyribose. Nucleotides are phosphoric acid ester of nucleoside. Phosphate combined with sugar molecules at its fifth carbon atom. However, phosphate linked to third carbon atom and rarely second carbon atom is also known. The number of phosphoric or the phosphate residue may be up to 3 in free occurring nucleotide. On the basis of pentose sugar present, nucleotides are distinguished into ribonucleotides, and that is ribotides, and deoxyribonucleotides or deoxyribotides. Being phosphorylated form of nucleoside, they are also called nucleoside phosphate. The various nucleotides are adenylic acid, that is adenosine monophosphate or AMP, deoxyadenylic acid, that is DAMP, guanylic acid, that is guanosine monophosphate or GMP, then deoxyguanylic acid, that is DGMP, uridylic acid, that is uridine monophosphate or UMP, deoxythymidyllic acid or deoxythymidine monophosphate or DTMP, cytidyllic acid or cytidine monophosphate or CMP, and deoxycytidyllic acid is DCMP. Higher nucleotides. Nucleotides having more than one phosphate group are called the higher nucleotides. Higher nucleotides occur in free state. The second and third phosphate of higher nucleotide are attached against the force of repulsion between similarly charged phosphate radical. Hence, the bond attaching the second and third phosphate are high energy bond, much higher than the common covalent bond. A number of high nucleotide are known, that is GDP, GTP, CDP, CTP, then UDP, UTP, then ADP, okay, and ATP. Out of these, ADP and ATP are well known due to presence of high energy bond, higher nucleotide function as energy carrier. ATP is adenosine triphosphate. Adenosine triphosphate is formed of adenine, a purine, a ribose, that is pentose sugar, and a row of 3 phosphate radical attached to the ribose. The complex form of adenine and ribose is called adenosine. ATP or the adenosine triphosphate was discovered by Carl Lohmann in 1929 from mussels. Its functioning was found out by the Fritz Lippmann in 1941. Lippmann is also called father of ATP cycle. Now, who is known as father of ATP cycle? It is Lippmann. The second and third phosphate of ATP are attached by high energy bond. Okay, the bond have high transfer potential because they are held against great electrostatic repulsion. ADP plus phosphate ion plus energy gives the ATP reaction can occur in a cyclic manner of build up and breakdown reaction. It is an instant storehouse as well as source of energy. ATP is found in all the living cells. It is also mobile within the cell. Therefore, ATP can pick up energy where it is being liberated and supply the same where it is being consumed. ATP is called universal energy carrier as well as energy currency of the cell. ATP usually obtain energy during respiration. It provides energy to the cellular constituents for overcoming entropy and carrying out various activities like membrane transport, nerve conduction, muscle contraction, biosynthesis, microtubule synthesis, cytoplasmic streaming, cell movement, cell division, osmosis, heat, etc. The last phosphate bond is usually built up and broken for this purpose. The enzyme is ATPase. Breakage of this bond release 8.9 kilocalorie, that is 8,900 calories per molecule. Our older estimate was 7.3 kilocalorie per molecule. Similar amount of energy is required for its building. The second energy rich bond has an energy equivalent to 6.5 kilocalorie per mole. Now next we have is nucleotide of vitamins, that is coenzymes. Nucleotide, nicotinamide and flavin part of riboflavin, two B complex vitamins function as nitrogen bases. Singly or in combination with other nucleotides, they produce important coenzymes which take part in oxidation reduction reaction. These coenzymes are NAD, that is nicotinamide adenine dinucleotide, NADP plus, that is nicotinamide adenine dinucleotide phosphate, FMN, that is flavin mononucleotide, and FAD, that is flavin adenine dinucleotide. Another nucleotide of vitamin is coenzyme A. It consists of adenine, ribose, 3-phosphate, pantothenic acid, and mercaptoethylamine. Coenzyme A function as acyl group carrier, for example, acetyl coenzyme A. Next, they may occur in free state but are biologically active in contact with specific apoenzyme. The enzyme formed from them are dehydrogenase. The coenzyme oxidize different chemicals by removing hydrogen from them. The reduced coenzyme have hand over their hydrogen to other chemicals. Now, what are the various functions of the nucleotides? Functions include they are building block of the nucleic acid or ribonucleotides of the RNAs, while deoxyribonucleotide produced DNA. Cyclic AMP or adenosine monophosphate function as second chemical messenger in many hormone controlled chemical reactions. Cyclic GMP guanosine monophosphate is a functional is in the calcium ions or the calmodulin mediated chemical reaction. Higher nucleotide behave as the energy carrier. ATP is known as energy currency of the cell since it contains high energy phosphate bond which are built up when energy is available and broken down when energy is to be utilized. Nucleotide produced by nicotinamide and riboflavin function as coenzyme, that is NAD plus, NADP plus, FMN and FAD of dehydrogenases or oxidases. Coenzyme A is produced by the reaction between nucleotide and pantothenic acid. UDP and ADP are involved in the synthesis of polysaccharides, while CDP and CTP are required for phospholipid synthesis. Now, next we'll move to the nucleic acids. Now, nucleic acids are long chain macromolecules which are formed by end-to-end polymerization of large number of repeated units called the nucleotides. Now, nucleic acids show a variety of secondary structure. There are two types of nucleic acid, that is deoxyribonucleic acid or DNA and ribonucleic acid or RNA. Now let's discuss the structure. DNA or deoxyribose nucleic acid is helically twisted double chain deoxyribonucleotide macromolecules which constitute the genetic material of all living organisms with the exception of the riboviruses. In Prokaryotes, it occur in the nucleotide and plasmids. This DNA is generally circular. In eukaryotes, most of the DNA is found in the chromatin of the nucleus. It is linear. Smaller quantities of DNA are found in mitochondria and plastids, that is organelle DNA. It may be circular or linear. Single-stranded DNA occur as genetic material in some viruses. For example, coliphage or the 5 into 174 virus. DNA is the largest macromolecule with a diameter of 2 nanometer, that is 20 Armstrong, and having a length in millimeters. It is a long chain polymer of several hundred thousand of deoxyribonucleotide. For example, 4.7 billion base pairs of E. coli and more than 3 billion base pairs of human being. A DNA molecule has two unbranched complementary strands. They are spirally coiled. The two spiral strands of DNA are collectively called DNA duplex. The two strands are not coiled upon each other, but double strand is coiled upon itself around a common axis like rope or spiral staircase with base pairs forming steps, while the backbone of the two strands form a railing. Due to spiral twisting, the DNA duplex come to have two type of alternate groups, major and the minor. The one turn of 360 degree of the spiral has about 10 nucleotide on each strand of DNA. It occupies a distance of about 3.4 nanometer, that is 34 Armstrong, that is pitch of DNA is 34 Armstrong, so that adjacent nucleotide or their base are separated by a space of less than 0.34 nanometer, that is 3.4 Armstrong. A deoxyribonucleotide of DNA is formed by cross-linking of three chemicals, that is phosphoric acid H3PO4, then deoxyribo sugar, that is C5H10O4, and nitrogenous base. Four types of nitrogenous base occur in DNA. They belong to two groups: purine, that is 9-membered double ring with nitrogen at 1, 3, 7 and 9 position, and pyrimidine with 6-membered ring with nitrogen at 1 and 3 position. DNA has two types of purines, that is adenine or A and guanine or G, and two type of pyrimidine, that is cytosine C and thymine T. Depending upon the type of nitrogenous base, DNA has four kind of deoxyribonucleotide: deoxyadenosine 5 monophosphate, deoxyguanosine 5 monophosphate, deoxythymidine monophosphate, and deoxycytidine 5 monophosphate. The backbone of DNA chain or strand is built up of alternate deoxyribose and phosphoric acid group. The phosphate group is connected to the carbon fifth of the sugar residue to its own nucleotide, and carbon third of the sugar residue of the next nucleotide by the phosphodiester bond. That is, hydrogen of the phosphate and OH group of the sugar are eliminated as water during each ester formation. At one end of DNA strand, last sugar has its 5 carbon free, while at the other end, third carbon of the first sugar is free. They are respectively called the fifth carbon and third carbon ends. The phosphate group provide to the nucleic acid because at least one of its side group is free to dissociate. Nitrogen base lie at right angle to the longitudinal axis of DNA chain. They are attached to the carbon atom 1 of the sugar of the glycosidic bond. Pyrimidine is attached to deoxyribose by its N atom at first carbon position, while purine does so by N atom at the ninth position. The two DNA chain are anti-parallel, that is, they run parallel but in opposite direction. In one chain, the direction is fifth carbon to third carbon, while in the opposite one, it is third carbon to fifth carbon. The two chains are held together by hydrogen bond between the bases. The adenine and a purine of one chain lies exactly opposite to the thymine, a pyrimidine of the other chain. Similarly, cytosine is a pyrimidine and it lies opposite to the guanine, okay, a purine. This allow a sort of lock and key arrangement between the large size purine and small size pyrimidine. It is strengthened by appearance of hydrogen bond between the two. Three hydrogen bond occur between cytosine and guanine at position first carbon and second and sixth, first carbon, second and sixth carbon, sixth and second carbon. There are two such hydrogen bond between the adenine and thymine, which are formed at the position first carbon, third carbon and sixth carbon and fourth carbon. Hydrogen bonds occur between hydrogen of one base and oxygen of nitrogen of the other base. Since specific and different nitrogen base occur on the two DNA chain, the later are complementary. Thus, the sequence of say adenine guanine cytosine thymine cytosine adenine guanine of one chain would have a complementary sequence of thymine cytosine guanine adenine guanine thymine cytosine on the other chain. In other words, the two DNA chain are not identical. It is because of specific base pairing with a purine lying opposite a pyrimidine. This make the two chain 2 nanometer thick. A purine base pair will take it thicker, while a pyrimidine base pair will make it narrower than 2 nanometer. A larger size purine therefore lies opposite to the small size pyrimidine, A opposite T and C opposite G. This specific base pairing make the two chain complementary. Sense and anti-sense strand. Both the strands of DNA do not take part in controlling heredity and metabolism. Only one of them does so. The DNA strand, which function as template from RNA synthesis, is known as template strand, that is minus strand or anti-sense strand. Its complementary strand is known as non-template strand or plus strand or sense or coding strand. The later name, that is coding strand, is given because by convention DNA genetic code is written according to its sequence. That is, the RNA in is transcribed on the third carbon to fifth carbon, that is minus strand or template or anti-strand of DNA in the fifth carbon, from the third carbon to fifth carbon direction. And the plus strand of DNA is that coding strand which carry genetic information but is non-template. The term antisense is also used in wider perspective for any sequence or strand of DNA or RNA which is complementary to the messenger RNA. Next is denaturation or melting. The hydrogen bond between the nitrogen base of the complementary DNA strand can break due to high temperature, low or high pH. The phenomenon is called denaturation or melting. Since adenine and thymine base pair has only two hydrogen bond, the area rich in adenine and thymine base pair can undergo easy denaturation. This is called low melting area. The area rich in guanine and cytosine base pair is comparatively more stable because three hydrogen bond connect the complementary nitrogenous base. DNA strand separate by melting can reassociate and form duplex. The phenomenon is known as renaturation. Palindromic and repetitive DNA. DNA duplex possess area where sequence of the nucleotide is same but opposite in the two strands. These areas are called palindromes or palindromic region. For example, MADAM, madam, and if you read the madam from backward also it will be same. So sometimes the bases, okay, the bases they are written such that when they are read from front and when they are read from back the sequence is same. So this happens on the two strands. So in one strand the sequence is same from the left to right, and from on other hand the sequence will be same from right hand to left hand. Okay, so such a sequence is called palindromic sequence. The exact significance of these short of arrangement is not known. Now, function of DNA. DNA is genetic material which carry all the hereditary information coded in the arrangement of the nitrogenous base. It has the property of replication, that is autocatalytic function, essential for passing genetic information from one cell to its daughter or from one generation to the next. Crossing over produces recombination. Change in the sequence and number of nucleotide produce mutation. Mutation are the fountainhead of all variations and formation of new species. It gives rise to RNAs through the transcription, that is heterocatalytic function. DNA control the metabolic reactions of cell through RNAs and the RNA directed synthesis of the protein, enzymes and other biochemicals. Differentiation of various body parts is due to differential functioning of specific part of DNA. Developmental stage occur in the life cycle of an organism by an internal clock of DNA functioning.